Hello, my name is Joshua Gao, and today my presentation will be over the four major microbes we use throughout our labs throughout this course. Um, yeah. For the introduction portion, today we'll be going through and reviewing the four main microbes and pathogens we use throughout this course. Those being Ecentric E. coli or E. coli, Staphylococcus epidermidis, which I will just call S. epi, and then Micrococcus and lutis, which I will just call M. lutis, and Seriata markinsons, which I will call S. mark. We will also be going more about their backgrounds. We will also cover the effects the bacteria have and whether a treatment is applicable, and further research that is done with it or on it currently. As we look at our first microbe, E. coli, we can begin the presentation. So, infamously known as E. coli and being in Chipotle's lettuce throughout the late 2010s, the National Library of Medicine says that this bacteria was first discovered in 1855 by Theodore Escherichia, and it was also a gram-negative bacterium. While everyone may know it as a dangerous and harmful bacteria, that isn't the necessarily full truth. Only a handful of these bacteria are actually truly harmful, specifically the strain of 0157H7, which we will just go and call H7. The bacteria E. coli lives in both humans and animal guts. These gut microbes can be seen as benign and or harmless. And to the right is a picture of the strain of H7 E. coli. As we further talk about E. coli, we can see that once the infectious strain of H7 was found in North America and other major countries, it began to spread. The two major ways of transmission was through food and water, but also found through human contact. The first case of this pathogen began in 1982 and lasted 2 decades until the study closed, concluding around 350 patients that reported having E. coli. If you contract E. coli, you may be asymptomatic, or worst case scenario, death. Although it was found that the majority of patients experienced diarrhea with no other symptoms. While there may have been multiple attempts at making a vaccination, no success has been shown. On the flip side, there are antibiotics that have been found helpful in relieving symptoms. But still, with no vaccination created and still pursuing one, scientists have learned to use E. coli to produce and test protein production, turning around the disease. And on the right is a picture that shows the Kirby-Bauer method, where E. coli is spread over a plate, and then there are discs that are filled with antibiotics to see which disc is the strongest against the pathogen, which we did as well in our lab. As we move on to our next microbe, S. epi, this bacteria also shares some characteristics to E. coli, in the sense of being gram-negative, generally harmless, and it uses the Kirby-Bauer method as well. It is found on the skin and lives there waiting to enter the body. However, it isn't that easy for the bacteria to enter. The body produces natural antibiotics to fight it off, and killing it before it gets to the bloodstream. Once S. epidermidis is inside a host, there are multiple possibilities of how the infection can manifest. Unfortunately, there is no cure for this pathogen, but treatment can still be given through an IV. As we talk more about S. epi, if a host has an ever severe case, then it should be known that the whole area of infection is to be cut off, meaning that prosthetics may need to be given. For example, if the infection is in your wrist due to a skin tear, then it may be needable. Worst case scenario would be to cut off your hand for a prosthetic, but catching on it early would be the best case scenario. And with the pathogen mainly affecting the skin and the body, there weren't many solutions to the treatment. But as technology advances, we fight S. epi head on. Micrococcus lutis. This bacteria was first founded in 1928 by Alexander Fleming and has the meaning of yellow spheres that are microscopic, which is the literal name of M. luteus. It is a gram-positive bacterium, and the pathogen has the normal living environment on soil, water, dirt, and the human skin. And it also has the effect of producing the odor of what sweating smells like. So every time you sweat or produce the sweat, this is the reason why. As we dive more down into M. luteus, the easiest way to be exposed to M. luteus is through poor hand washing techniques. Once contracted, the most common symptoms are fever, chills, and possibly hypotension. While there's no vaccine for this bacteria, treatment is available through various medicines added together. And there is a now wash your hands to support being clean and staying healthy. While the pathogen is generally seen on agar plates and used for lab cultures, there are other methods to recognize the bacteria with the MALDI-TOF or the Corkia sp, also using the streak plate method, which we use throughout our course. Other factors have also pointed into the direction of biodegradation, so turning around this bacteria for the better use of society today. The S. marcansons will conclude the major bacteria throughout this presentation and the course. It was founded by an Italian pharmacist in 1819 by Vizio. The pathogen is mainly prevalent in hospitals that focus on critical care and pediatric units. Poor hand hygiene is also the number one reason due to its growth. So once the pathogen has entered the host, numerous effects can take place dependent on how it enters the host. A large portion of the causes or cases take place in the respiratory tract, then spread throughout the body, and they can also be found in the UTI, epidermis, and other main functions throughout our body. As we go on to the next slide reviewing S. marcansons, it's important to know that some of this information was used from a case study, and that can be found in my references. While S. marcansons is harmful, it was also found to be treated due to its sensitivity to iminopen and didn't reappear for 6 months, meaning they administered this microbe to hosts and patients, and then were able to successfully eradicate it from them. And then they even went as far as finding a vaccine for malignant pleural infusion, which is also one of the effects S. marcansons has. And another reason why we use S. marcansons is because it's easy to see growth due to red colonies and the streak plate method. And even today is commonly used for educational purposes, such as this class. Now for the conclusion. So after reviewing these pathogens, it's safe to say the best way to prevent any of these is through proper hand washing techniques and overall cleaning, especially since most of these are used for educational purposes. However, this presentation can inform you with the microbes you used, the effects they have, and why you should protect yourself. Also just giving more of a background on how they can be used or what they were used for, or context in all solutions. Here are my references.